All right, virtual students. I wanted to make this video for you to talk a little bit about the DNA probe lab uh, that we're going to be doing in class. You guys are going to do this at home as well. It's not incredibly difficult, but it is kind of difficult to demonstrate, especially when I'm trying to show the face-to-face kids at the same time. So this video is really for you guys to have an idea of what's going on with this. So I'm going to post a lab handout that has all the directions on it, and it's just going to be slightly modified for you guys. Basically, we've just finished up, or we're finishing up the unit on DNA, and we know a few things about DNA. First of all, we know that DNA has base pairs, adenine and thymine. The A's and the T's always go together, and guanine and cytosine, C's and G's, will always go together. So you need to keep in mind those base pairs when we're doing this lab, okay? So what happens is we have mutations or mistakes in DNA, and most of the time they're very, very minor. They happen really regularly, and you never even know they happen. But other times they are more damaging or more severe, and scientists have the ability to go into your DNA and basically cut out the bad portion and replace is going to basically, help overcome those genetic abnormalities. The problem is the number or the sequence of DNA is so very basic, very primitive one-cell organisms have 4 to 5 million base pairs. When we get up to humans have over 3 billion base pairs. So finding the location where the gene or the DNA sequence is out of order is a problem. So to do that, scientists use DNA probes. So what a DNA probe is it's just a piece of DNA, okay? It is, it kind of depends on the length. It's going to be longer than what we're simulating in class, obviously, but the length varies. But basically what it is they'll have this section of DNA. So for this section of DNA, we have I can't read backwards. We have ATCCGA. This yellow down here represents a fluorescent dye that the scientists attach to their DNA probes. So what they'll do is they'll put this into the body, and this particular DNA probe is going to attach to the complementary sequence. A's and T's will go together, and C's and G's will go together. Wherever it attaches for the entire sequence, it's going to stick there, and then the scientists can see this fluorescent dye, and they can go in there and they can cut out that piece because they'll then know the location. Okay? So on the lab sheet, you're going to be given 5 DNA sequences. This only has 2. Okay? We're going to do this on a sheet of graph paper for the face-to-face. If you have graph paper at home, that would be great. It would make it very much easy for you to do. If you don't have graph paper, then you're going to have to improvise. You're going to have to do this on a sheet of paper and create your own squares, kind of like what I've done here on this little piece of poster board. Yours obviously doesn't have to be this big, but you're going to have to be able to put these DNA sequences down on a sheet, and there's going to be 5 of them on your assignment. And then you're going to be given a DNA probe. So what we're trying to do is we're trying to identify individuals that match up with this DNA probe. Okay? So what we're going to do is we're going to take this and we're going to start looking at our matching sequences. So you can see A and C do not go together. A and G don't go together. A and T do, so it goes together there. T and A, C and G, C and G, G and C, and A and T. So right here, this piece of DNA is where this probe would attach, and it would identify this individual. Okay? Then we're going to take this DNA probe and we're going to go to the next sequence. A and G does not go together. A and A, no. A and A, no. A and T, T and C do not go together. So that's not it. So we're going to go on down. A and C, that's no good. AG, AG, AT go together, TA go together, CG go together, CG go together, G and A and T. So right here from T down to T, that's going to be our complementary DNA sequence. So what you'll do is, wherever your complementary sequence is on your paper, if there is one, you'll just circle it and you'll identify the individuals. That's the basic premise of the lab. The rest of the stuff on the lab, the questions that go along with it, those are all pretty self-explanatory. Just lay down your code, and when you write your code down, when you copy it down on your paper, on your graph paper, or whatever you copy it down on, pay attention to what you're doing. When people mess this up, it's because they copy the code down wrong. They copy it down incorrectly from the beginning, which causes it to affect the results. So you'll make your sheet like the poster, you'll make your little probe like this, and you'll basically just slide it along and find the complementary spots on the DNA sequence and use that to answer the rest of the lab questions. So that gives you kind of a basic idea of what I'm looking for. Hope this helped you guys out. And if you have any issues or any problems, as always, just send me a message and I'll see if I can help you out. So good luck with this. Have a good day, and I'll talk to you later.